the mess is kind of organized back here. So, I'd like to thank you for coming today. We've been planning this for a long time. Here, retarded. Even working half the time. And we've done a lot the past year. We've had, a lot of good things, coming.
Hey guys, wait. He's going to give our little on-the-speed stuff. We'll have to crack deals in a little bit if we can get coffee a moment to get us going here.
I know, it's really hard to give me any time. I get it.
It's Kurt's fault.
Kurt, yeah.
Dude, give me the battery.
Alright. Much better. So, technology never works out for me. I work in computers. Does that work? All right. Woo! So, thank you all for coming today. We really appreciate it. We are really excited about being able to rent out the Burden Center. Obviously, this is a very nice complex, and it's well, you go. You can get a baby. The first time we came in here and looked at it, we were like, yeah, this is, if we could fill this place up, then we'd be really happy. We've done a pretty good job of filling it up. So, thank you all for coming. We'd like to, really thank all of our sponsors. Obviously, without them and you guys who are supporting us, we wouldn't be able to make things like this even remotely possible. So, thank you for that. JT, we want to thank him for doing a phenomenal job preparing all the jambalaya, excuse me. If you've never had it before, hopefully you enjoyed it because it's really good. We have two really great speakers up here today, Butch and Murray. They have a couple of different topics that they're going to be, kind of going into detail on, a little bit different angles on some very, very important things in the hobby. So hopefully it won't be, too exciting of a presentation, since I know this one is right now. I think that's all I wanted to say. Is there anything else? Yeah. Thanks again for coming out, guys. And I guess, which year's coming up first? All right.
Welcome, Butch.
Yeah, let's give him a big round of applause. He came from St. Pete to visit with us today.
Test? You got me?
Got you.
Alrighty. Alright, well, my name's Butch Ringelspaugh. I am from sunny St. Petersburg, Florida, which in a minute we're exactly located at. How many of you have been to Tampa Bay, around Florida? Okay, so we got some familiar people. But before I get started, I really want to thank the Bayou Reefkeeping Club for bringing myself out here, and Murray as well, joining me. It's an honor to have you guys reach out to me, and we're interested in what I do to talk to you guys about, and hopefully bring up some new ideas for you and some exciting discussion. So, the title of my talk is Growing Pains of the Atlantic Caribbean Acroporids, and my aquaria-based approach to help them in a time of need. Let's see if my clicker's working here. Okay, so first we're going to talk a little bit about my background, a little bit about the corals that I'm most interested in and what's threatening them, what I'm doing with my research and methodology, and some research aquaria design and development, what I've done so far and what I plan to do in the future. So there's my home. Born and raised in St. Petersburg, Florida. Grown up on the water, boating, snorkeling, scuba diving since I was 13, going down to the Keys and the Bahamas and the Caribbean. So I've been totally engulfed into the ocean and marine science, and that's what's really gotten me interested in what I do now. I am the curator of exhibits at a public aquarium called the Pier Aquarium, which is in downtown St. Petersburg in this iconic structure called the St. Petersburg Pier, which is a big upside-down pyramid about a quarter of a mile out into Tampa Bay. And at the same time, I'm also a graduate student at the University of South Florida College of Marine Science, which is just down the road from the pier. And that's kind of a segue into what makes St. Petersburg very special and what's kind of helped me become more interested in marine science. The aquarium here, our mission is representing what's called the St. Pete Ocean Team. The St. Pete Ocean Team is a consortium of about 14 or 15 marine science organizations that are all located there on the waterfront in downtown St. Petersburg. We have about 1,500 marine scientists that work out of downtown St. Pete. We're kind of at the level of Woods Hole Oceanographic Institute and Scripps out in California. We have groups ranging from NOAA National Marine Fisheries Service, the United States Geological Survey, USF. We have a company called SRI International, which they are a technology-based group that produces oceanographic equipment. You might know them because they were the ones responsible for designing and inventing the mouse for your computer. So there's some pretty incredible stuff that happens in downtown St. Petersburg. And what we do at the aquarium is we take that great work and pass it along to the public and let them know what's happening in our backyard and how these marine scientists are benefiting the global marine environment. So a little bit of introduction on the corals I'm most interested in, and a little background on why I became so interested in them is I've been going down to the Keys since the 80s and into the Caribbean, and these corals just stood out to me. The elkhorn coral and staghorn coral, beautiful reef building species. The first time I saw them and it really struck a note with me was when I was about 14 years old, snorkeling in the Dry Tortugas National Park. And I was helping out with a nurse shark research program down there. And I was first interested in sharks, but then these corals kind of stole the show for me, and I've been focused on them ever since. And since the 80s, of when I've been going down to the Keys, I've seen the water quality degrade over that time. I've seen the corals kind of step back and decline quite a bit. And so after a while, once I got through college, I got my bachelor degree at University of South Florida, St. Petersburg in Environmental Science and Policy. And I was working at the Pier Aquarium and the Florida Aquarium in Tampa. And I was working at them both for several years. I became a coral biologist at the Florida Aquarium, and I got to participate in lots of coral restoration projects with them, one being Seacor. Have you guys heard of Seacor? Who's heard of Seacor? So Seacor stands for Sexual Coral Reproduction. And there's a group that goes down once a year to specific sites, the Puerto Rico reef habitat there, the Curacao, and the Florida Keys, for when elkhorn coral and staghorn coral are predicted to mass spawn. And they collect the larvae, or the gametes, mix them up, create larvae, settle those larvae in their aquaria so that they can be reintroduced out into the field once they grow beyond a certain point. So I started working more and more with these species of coral. So Acropora palmata and Acropora cervicornis, those are the only two true species of Acropora throughout the entire Caribbean region. In the Pacific Ocean, which is you're most familiar with, I'm sure, working with the Acropora species from there, there's hundreds of species, lots of potential for hybridization. Speciation and species identification is extremely complex, and it's very difficult to get them beyond just an Acropora genus down to the species. I know that a lot of you probably try your best to identify, but it's extremely difficult. In the Caribbean, there's only two of them, staghorn coral and elkhorn coral. And since 1980, they've died off by almost 95% through their entire range. They were once one of the most dominant species of coral through that entire range, responsible for building the majority of the reefs through the Caribbean. And since 1980, they've been knocked back by almost 95%. That's astonishing. That's happened in a very short amount of time, so it's a very alarming issue. So in 2006, they were listed as threatened under the Endangered Species Act, and since then there's been efforts to to research more and work out ways that we can conserve the species and not let them go extinct. The next coral, which is I've been extremely interested in, is Acropora prolifera. It's called fused staghorn coral. This is the hybrid between elkhorn and staghorn coral. It's not found in the fossil record. It's only been occurring relatively recently in the natural environment, geologically speaking. So the question that was posed is, why is this coral just all of a sudden occurring in this recent time? And so some have hypothesized that that might be a strategy for the parent species to survive changing environmental conditions and beyond. And this hybrid has become more prevalent, more robust, and so I'm really interested in its vigor and if it does play a role in the future of the reefs of the Caribbean. And the one thing about this hybrid within the ESA threatened status is they don't see it as its own species, because it's only an F1 hybrid. It hasn't been sexually reproducing on its own to be self-sustaining of its own species. So until that point, they won't put it on the Endangered Species Act unless somebody creates a great argument for why it should be. So it has not been listed as of yet. So this is elkhorn coral, Acropora palmata. Comes in a couple of different growth forms. And this is the coolest Acropora in the world. This is the largest Acropora species in the world. It can grow 6 to 8 feet in diameter, grow for several hundred years in a single colony, and they grow in incredibly active environments. So this is the more branching form of it. You can see where it gets the elkhorn coral name. This is the more robust form, where it grows in more of the shallow habitats and the more physically active environments, higher currents, higher waves. In fact, they deal with conditions like this. This is off Puerto Rico. This is a famous surf spot. And the reason these waves are breaking there is the reef is built by the elkhorn coral. And they can deal with this type of high energy environment because they're so robust and they're built structurally in a way that they can deal with it. This is Acropora cervicornis, so this is staghorn coral. It's more of the branching morphology. And then this guy is the fused staghorn coral. So it takes a trained eye to really differentiate this to that. And the only thing that is not a problem with it being listed as an endangered species is it pretty much falls under that listing anyways, because any average person out in the water, even almost every average scientist out there, is not going to know the difference between this and the staghorn or elkhorn coral. It comes in a couple of different growth forms. Some is more bushy, some is more branchy. And this is another one there. And the one main way you can tell the difference between staghorn and fused staghorn coral is fused staghorn coral, the branches come out in more of a cluster, more of like a 45-degree angle, where staghorn cervicornis coral branches out at almost 90-degree morphology. This is a very interesting specimen of fused staghorn coral. I was just talking to Murray about this last night. This guy is growing in the mouth of the Miami River, and one of the most major shipping channels in the world. It's growing within about 1 foot of the surface. It has survived the 2010 cold snap without a problem. During low tide, it's maybe an inch or two below the surface. And let me tell you, the environment that it lives in is absolutely terrifying because of all of the waste and crud that goes into that river and deals with all the shipping traffic. There's goats in the water from all of the practices they have in Santeria down in Miami. And this coral is doing pretty darn well. And a gentleman, Colin Ford, down in Miami, that has the company Coral Morphologic, and I think it's shifted to another name now, Aquascapers. He alerted me of this and went down and checked out this coral, and we're trying to work out a permit to take a couple of fragments of this coral to bring out to a nursery to grow out for future research. There is a planned deep dredge project that is supposed to happen in Miami, which could essentially be certain death for some of these corals living in that area. So we want to act kind of quickly and salvage this special coral because obviously it's extremely robust, and also with these corals, they mass spawn, they produce millions of larvae. There's probably been thousands to millions of larvae that have floated over here and landed on this structure, and only this coral ended up surviving. So the combination of genetics that allowed it to survive is incredible. This is an entire area of reef dominated by that hybrid coral. So they do have the potential for building reef. So just to give you a little bit of more of a broader picture of where these corals are from, we've got the Caribbean here. These corals grow all throughout this entire region, and then up into the Florida Keys, and then they stop about right here. We have a nice old loop current that comes up through right in this space here and loops around the Gulf. And you guys probably heard of the Flower Garden Banks, which is just off the coast of Texas, and that's really the only true coral reef of the Gulf of Mexico. We have coral in the Gulf of Mexico, but they don't actually build reefs. That reef there actually has reef-building corals, and it's a very unique habitat. And that larvae from all those different corals have just floated through this strait here and have managed to find a nice habitat up in the upper portion of the Gulf. And so remember the oil blowout that occurred right up in the northern part of the Gulf. We were saved down here from any effects. A lot of people were worried that that oil was going to come down to the Keys. That loop current actually had a phenomenon where it pinched off and just looped off and kept it from coming down to the Keys and out into the Gulf Stream and out. This is a little bit of more of a labeled map here. Curacao, that's where I was talking about, where they do the Sea Corps program, Puerto Rico. And so we've got the bigger world map here. And just a comparison of the Pacific Ocean, where you've got all your Indo-Pacific corals, compared to the little basin where the Caribbean is. So there's a lot less species diversity, a lot less coral mass in the Caribbean, because it's just a smaller basin, but we see it as kind of a bellwether of what may occur in this larger tub of water that we call the Pacific and Indo-Pacific. So the way these corals are populated, like I said before, they're broadcast spawners. This is a photo of an elkhorn coral about to spawn. All these white dots are egg and sperm bundles. This is a hermaphroditic coral, so they contain both male and female gonads. They create egg and sperm bundles, and then at one time during the year, they will all synchronously release those egg and sperm bundles. They float up to the surface, into the current. They mix together with other genetic strains of that species. They drift for a few days and will settle down and grow into a new coral. And they can spread through the Caribbean that way. There's a little bit of a closer look at those egg and sperm bundles compared to the ones without. And so that's what we were looking for. We were looking for the polyps that were teeing up. They'd tee these things up before they release them, and we would catch them as they were coming up. And there it is, a reverse snowstorm of egg and sperm bundles. And then when they settle, they have a primary polyp like this little guy, and we've got some zooxanthellae starting to colonize. And they grow a little bit larger. This is a juvenile elkhorn coral. A little bit more mature. And then they grow out to these big, amazing colonies. And so now to the threats of why they've been knocked back by so much. This photo was taken at Carey's Fort Reef in the Florida Keys in 1960. And then again, the same spot in the 1980s, and that reef was totally decimated. You go down to the Florida Keys now, and you're lucky if you see one of these two species growing. One of the primary threats was disease. And recently there was a paper that was published that linked a human-derived bacteria to white band disease that was occurring in the Keys. Down below, this disease, this is called hyperplasia, and this was actually, it's not a banned disease, but actually extra calcification. They hypercalcify. And I wanted to show this photo because this colony here is infected, this one is not. They're right next to each other. And this just goes to show you that there's, beyond just the individual species, the species is made up of a lot of different genetic strains. And so certain genetics might do better than others in certain conditions. And obviously this strain is probably resistant to whatever this colony is inflicted with. Sedimentation is a big problem. And that's from runoff and dredging and whatnot. That when land is developed and you're pulling up all these big parcels of land, knocking down the trees, developing the land, all that sediment gets loose, goes in the river system, and goes out to the reefs. Golf courses are a big problem in the Caribbean, not only from them denuding the island and letting loose all of the sediment, but also because of all the fertilizer that they put on the golf course on a regular basis. And the sediment can just smother corals already existing, but then it also smothers the habitat for the new little larvae to settle and grow into bigger corals. And that's really the key, is the sexual reproduction. These corals need to recruit and be able to settle their larvae on the reef. And if there's not that real estate to do it, that's not going to happen. This guy is a diadema urchin, long spine urchin. They're very important to the reefs of the Caribbean because they keep the algae in check. They groom the reef so that there's that habitat, that real estate for the larvae to settle. And in 1983, this urchin species died off by about 98%. There was a systematic die-off throughout the entire Caribbean. There was a disease that all of a sudden came about and decimated these urchins. And now they're starting a recovery. There's a gentleman that I'm sure you've heard the name of, Martin Moe. He is doing his form of coral restoration by aquaculturing this urchin to outplant on the reefs. So this just goes to show you how extensive our watershed is and how we all impact the reefs of the Keys and the Caribbean. And this is always opens everybody's eyes. And, you know, you could be all the way up in Montana and have an effect on our reefs down here by what you're putting in the water. And you just think about, as that river's flowing down, how much stuff is accumulating all the way down into the Mississippi until it reaches the Gulf of Mexico. And we've got this huge dead zone in the Gulf of Mexico because of all the nutrients that end up in that basin. Another problem is ship groundings. This is the wellwood that ran aground on Molasses Reef in the Florida Keys in 1984 and took out almost 75,000 square meters of reef habitat. If you've heard of the Coral Restoration Foundation run by Ken Niedermeyer in the Keys, growing out the staghorn coral, his first permit to outplant was to outplant on Molasses Reef to replenish the stock of coral that was demolished by the ship. Nice little anchor laying across the coral that can flip over massive corals that have been growing for 100 years or so. Trash. Can't tell you how much trash I pick up for the reef when I'm down there diving. Rope, bags, nets, all sorts of stuff. And kind of an overlooked issue is African dust. This is a satellite image of a dust storm blowing off of the western continent of Africa. And this dust actually ends up in the Caribbean. And there's some scientists that are studying how this might affect the health of the reefs by its incidental introduction of pathogens that ride these little dust particles all the way into the Caribbean and then can inflict a disease epidemic in the Caribbean. It's also a source of iron. Cyclones. That's a natural occurring phenomenon, and they can wreak havoc on reefs. It's naturally, you know, break some branches off the coral, float along and regrow. In a perfect world, that's great, but now that they've got all these other threats impending on them, they've got to deal with these cyclones coming by and knocking them back even further. It doesn't help their recovery. We have our pests. This is a fireworm, and this is the guy that I think caused hysteria within the reef club or reef hobbyist community. Where I remember years ago, everybody was afraid of all these bristleworms in their tank, and they were thinking it was this species that actually ingests tissue on the coral. But those bristleworms that we typically had were just the detritivores and the ones that eat the junk that settles on the bottom or the dead fish or whatever. These guys are much bigger, and they actually swallow the branch of the coral and digest the tissue while they're over the coral, and they back off and they leave a few inches of bare skeleton. So this is the guy that folks used to be worried about, but they're actually quite rare in aquaria. And that's, you don't even have to explain that. And then we have competition. So this is the fire coral, infamous fire coral. Millipora is the genus, so not our type of Millipora that we grow from the Pacific, the Acropora millipora. This is the fire coral that is opportunistic. And when, say, the elkhorn coral dies off a little bit, this guy will colonize the denuded skeleton and then start to push the live elkhorn coral out of house and home. This is another one, Palithoa caramorum. This is a matting zoanthid that will take over expansive territory. So new corals can't settle. And then we get to bleaching from thermal stress, and that happens with cold water scenarios and warm water scenarios. And then the combination of thermal stress with the impending threat of ocean acidification. So with more CO2 going into the atmosphere, the more it goes into the ocean, and that CO2, like many of you know, as it gets into your tank water, it can drive down your pH. And since the Industrial Revolution, we've experienced a 0.1 downgrade on the pH scale, which is huge. Most people, when we talk pH, it doesn't sound like that big of a deal between 8.1 and 8.0, but we're talking a logarithmic scale. We're talking like the Richter scale for earthquakes. Somebody experiencing an 8.0 earthquake compared to a 7.0 earthquake, it's a big difference. So any change on that pH scale, just by that 0.1 unit, is a major change. And so scientists are predicting that we might end up down closer to 7.9, 7.8 on the pH scale within the next 100 years or so. And that's a huge problem. I know a lot of folks at home with their reef tanks, their pH might fluctuate from 7.8 to 8.0, and you don't really notice an issue, but you don't know what's going on inside the organism. You don't know what's going on inside the coral, what type of structural issues they're having, or if they're producing their gametes for sexual reproduction or whatnot. Oh, going the other way. So, I really wanted to develop a project that would be as impactful as possible, that I could also, on the side, get my master's and PhD with. And so I was really interested in these corals. I was intrigued by the impending threat of climate change and what I could do to help the collective effort to conserve the species. And so I designed a multifaceted project. At first, I just wanted a master's out of it and just a basic project, and then it just became much larger. And so NOAA National Marine Fisheries Service funded the project initially with the caveat that they wanted me, there's one specific part that they wanted me to cover for them. And that was doing a review of current methods and practices for keeping these corals in captivity. There was a group that they funded that did a review of practices and methods with the groups that do the nursery, the offshore nursery efforts, so that they could put that into one document and provide it to other interested practitioners so that they didn't have to beat around the bush on how to figure out how to successfully cultivate their own nursery. So this is kind of the same thing. And my goal is to speak with all the individuals that have these two species of coral in captivity, which there's only a handful because they're highly protected. You're not going to see the species in the local fish store. Only public aquaria and marine research institutions will have these species. And the elkhorn coral is one of the most difficult corals to keep in captivity. It's been kind of the holy grail within reef aquaria within the commercial side of things. So that's the first part. And then I want to ultimately design and construct my own experimental aquaria so that I can replicate future conditions and compare how that's going to affect those three species. And I primarily wanted to look at the hybrid vigor 3 with that fused staghorn coral. So the benefit of using aquaria for research and coral conservation is, one, you don't need to go into the field except to get your initial stock of coral. So you can have your own experiments set up in your tanks. You don't have to worry about Mother Nature and all the expenses of going out there and being at the mercy of the sea. We can back up, you guys know you can back up in situ, which when I say in situ, I mean in the ocean. Ex situ means in the lab in our aquaria. So we can back up those ocean stocks with what we're growing in our tanks. Research can be expedited. I can look at a scenario that is predicted to happen 10, 20, 30, 40 years down the road rather than going out to the field just watching it day by day. And you can get to what potentially will happen so we can elicit change in our management much quicker. And of course, it's an effective educational tool. So one big way to help the whole situation out is getting everybody on board with what the problem is, enlightening them. There's this issue going on that will directly affect each and every one of you no matter what, and that we need to do something to protect these very important species of coral. So then the next, of course, once the aquaria is designed, is successfully acquire, transport, acclimate, and culture both of these species, which will be a task in hand on its own. And then address any pitfalls, and then do the climate fitness comparison. So starter with the survey of best practices and methods. I developed an extensive survey which involves from what size tank to what type of filtration you use, the water, your water quality regimen, what you use to keep up with your various nutrient levels, whatever. And I've distributed it amongst the Coral List and Aquatic Info listservs, which is kind of like one big forum for public aquaria and research scientists. And then I've been traveling to all the different locations with my own light and flow meter to get actual numbers for very critical parameters, especially flow. It's one of the most undermined parameter, in my opinion, in aquaria, where I'll say, how much flow do you have in your tank? I say, oh yeah, a lot. My polyps are blowing all over the place. But then as soon as I stick my little flow meter in there and show the number that correlates to what you're seeing, it's much different than what these corals actually experience out in the wild. And so I'm just getting to the bottom of what the actual numbers are. And like I said, there's not many people that have these corals in the world. This is it, pretty much. There's maybe a couple more out there. We have from the University of Florida Tropical Aquaculture Lab, we have a few big aquariums that you'll recognize. Nova Southeastern, they've got a big effort down in Fort Lauderdale now for staghorn conservation. Erasmus, that's the University of Miami. Georgia and Steinhardt just got a hold of some of these corals. And what's great about this survey is it shows all the different ways that you can successfully keep these corals. There's not just one written regimen for how you can keep these corals. There's folks that have an outside nursery with just shade cloth for their lighting supplementation, and this is down at Nova Southeastern. They use saltwater wells for their saltwater production. They pull out of a saltwater aquifer, and it comes up with all sorts of sulfides and low pH, and they have to treat that before they use it. So that's one method with natural light. Then others use artificial light here at the Florida Aquarium, but they use water from the Gulf of Mexico that they barge in. And then others have their artificial seawater that they produce with their RO unit and their instant ocean. And they'll mix that with artificial light scenario as well. And then another unique instance is with the Smithsonian. They have the largest elkhorn coral in captivity I've ever seen. And their primary filtration method is with an algal turf scrubber system. And they are extremely successful at what they do. And it's just another avenue of how you can be successful in keeping these corals alive. So there's not just one way. So my goal is to create a cookbook on what's been done, what's been successful, what hasn't been successful. So then that can be a resource for future aquarists and scientists so that they can just get down to the bottom of things. Yes?
So at what point do you broaden the 15 or 12 to the hundreds of thousands of hobbyists to say, now let's see? Because as you know, the last 20 years, much of what's been learned has been in someone's living room.
Right. It's a good point. And Murray and I were talking about that last night. And you know, there's hobbyists all over the world that grow corals in captivity and are successful in it, and they're the ones that pioneered how to do it. And it's tough to convince the government who manage these resources to let hobbyists have the ability to grow these corals out and then outplant them into the wild. So what I'm kind of starting that movement, but you got to start somewhere.
Sure.
Where, since it's happening in zoos and aquariums right now, and marine science facilities, my goal is to start a species survival plan within zoos and aquaria, kind of like what they do with pandas and lions and tigers and that sort of thing, to have a certain genetic stock backed up. And in order to get just to that point, you have to have a very convincing argument on how that can occur. So by having these methods in place on how it can be done, and then have the ability to manage it, so you know that the corals that are in these tanks are only being grown in tanks that have corals from that region, and not corals that are from all over the globe. So they're very particular on where these corals are grown and what other influences might be. If there's certain bacteria, disease, zooxanthellae clade that's not known in that certain region, they want to be able to manage that. And they even separated the keys. Actually, I'll start back a couple steps. The Caribbean is separated into two parts. They have the Western Caribbean and they have the Eastern Caribbean. And they've even found out that there's total genetic diversity difference between the Western and the Eastern Caribbean. And then they've even found that in the lower, middle, and upper Keys, and then southern Florida. So there's a lot to think about on how to appropriately manage them. So that this is what I'm just trying to start with. We have the first effort in zoos and aquaria to have a species survival plan for a non-mammal. This is the elkhorn coral at the Smithsonian. That's the largest in captivity right now. There's two of them right side by side. And these guys were raised in aquaria. However, they were raised with Pacific coral, so they technically can't have the ability to release them out to the wild. And I'll get back to that in a little bit. So to my effort with the aquaria design, I developed an aquarium system that I wanted to serve two purposes with. One is I wanted to be an exhibit at our we're actually going to be developing a new public aquarium that we're moving to. So I wanted it to be exhibit worthy, but I also wanted it to be as functional as possible for a multitude of research potential. I envision not just being the only person to use this system for coral research. I want to be able to invite other scientists in to utilize what I've developed. So I'm kind of developing a clean slate that has a lot of potential for a lot of different things. So the aquaria is actually in a three-stepped fashion. And for the exhibit-wise, that way I can show what corals grow in which different habitats. And then we can get into talking about how sea level rise and fall occurs and how that affects the topography of reefs. But then I can also, within the same water chemistry, look at three different lighting and water flow regimes in the same tank. So the three corals I'm working with, the staghorn coral, they like a little bit deeper, a little bit less water flow. The hybrid likes a little bit in between, and the elkhorn coral likes the real aggressive water flow and a lot of light. So above each section there's a 1,000-watt metal halide. And so that way I can raise the light or lower the light, shade it, whatever, so that I can achieve the light levels that I want to achieve. Each tank is about 350 gallons. They each have I should say there's 2 tanks total right now. I have a 1.5 horsepower water pump, reflow water pump, providing the water flow. So I can achieve water flow of up to 1 meter per second up in this top level here. The reason I want to be able to manipulate the water flow with the light is looking beyond the ocean acidification problem. I want to be able to also look at the combination of thermal stress, the temperature stress, with the light stress and water flow. That's kind of the combination of what causes a coral to bleach in the wild. Is if you have warm water with a lot of flow and not much light, they'll do okay. But if you have stagnant water, when it's real nice when we want to go out diving, and it's in the middle of the summer, a lot of light, and it's warm, that's when they start stressing out. And so that way I can kind of fine-tune of where that threshold is, where they start to bleach at a certain water flow level with a certain light level and temperature level, and then also look at the ocean acidification factor. So this is as applicable as possible. So there's my lab over at the college. I've got these tanks sitting side by side. They're nice, pretty tanks so that they can go on exhibit. And they've got that nice 3-step design. So got that applicability for a multitude of research projects. There they are sitting side by side. And I wanted to be able to handle a lot of water flow. So I've got the spillway that goes the entire length of the tank, so I can throw as much water flow at them as I want to. And I've got about 5 1.5-inch drains going into a big pipe going into my sump. Just going step by step on the plumbing. Got a basic filtration setup, sump, filter sock, protein skimmer, calcium reactor, automatic top-off, reservoir, just a basic setup. Had some nice calcium reactors built for me by Next Reef, and these were not only just designed for doing the calcium reactor method, but also to use these for treating the water for my ocean acidification experiments, so that I can set up a certain water chemistry in those cylinders and dose it at a certain rate to the tank so that I can achieve the correct ocean acidification parameters in the tank. There's a lot of playing with that yet to go. These 2 round tanks, this is what I started my water stock with. These tanks were filled with live rock from the Florida Keys and instant ocean seawater, some fish from the Florida Keys, and I had cultured that water for about a year before I ended up moving them into my research tanks. And that was just to go through all the ups and downs of algae blooms and blooms and die-offs and get that nice stable water chemistry, because when I get these corals in my hands, I don't have a whole lot of leeway. If I lose these corals, then I have to go through the whole permitting process again, and it's a big setback. So I wanted to make sure I was completely ready. So the tank is nice and full. You see lots of water movement there. We've got our 3 big lamps up there. Adding the live rock from the other tank, and the water is all from those circular tanks. Got some nice little blue tank, Atlantic blue tangs in there. And then started to populate with staghorn coral. This is from Kent Niedermeyer's nursery down in the Florida Keys. And when I applied for my permit through the Florida Keys National Marine Sanctuary, I specified that I want the corals to come from his nursery, so I don't have to go out and fragment wild corals and bring them in, because that's a little bit tougher, and this is a little bit more environmentally conscientious. And these came off his line nursery. I've got them hanging off of airline tubing on their own little cords there. This is my salamander coral. You can see them hanging here. So this is my first stock. They're doing wonderfully. They're growing really well right now. So I'm just getting my stock built up. I still have I was permitted for 20 different coral colonies of each species with 4 different genotypes within those 2 species. So that's what they looked like out at Ken Niedermeyer's nursery. And I'm just about to get my first elkhorn coral in captivity. And that's going to be the first time Ken Niedermeyer's exported any of his elkhorn coral that he's growing in his nursery. I'm also recently decided to add on some extra corals species. The Florida Keys National Marine Sanctuary reached out to me and said, we've got a bunch of coral in a nursery down in Key West. Can you use it for your research? I said, yeah, absolutely. So I went down to their nursery and picked up a bunch of brain coral and star coral. This is a nice big Culpophilia brain coral. It's about that big around. It's doing phenomenally right now. We've got another type of brain coral that's called Diploria clavosa. This is a Montastria faviolata. And these guys came from a seawall in Key West, in the Truman Annex. And they were planning a demolition of that seawall. And all these corals were growing right there on the seawall vertically. And had these big, massive corals. So they decided that they would harvest those corals off the wall to save them from the demolition. And that's just what they did. And they hang them on the docks here. I don't know if you can see that there's a line there and a line there. And underwater there's a basket with these corals just sitting right in there. And they're doing great out there in the Key West Harbor. And I just brought these corals back maybe a month and a half ago. So I don't want to run over on my time. How am I doing? Okay. My plan for getting the corals, like I said, I wanted to get it from the coral nursery. Oh, what's happening? There we go. So wanted to get it from the coral nursery. That's another picture of Ken Niedermeyer's site. And I understand that the next MACNA is down in Miami, and there's some potential for getting people out there to check out the great efforts that Ken is doing. He has incredible success, thousands of staghorn coral and elkhorn coral, and he's been doing a great job of outplanting them onto the reefs. And the reason I wanted to get them from the nursery also is because wild fragmented corals do pretty poorly, especially with these 2 species, and specifically Cropra palmata. This was in Puerto Rico. We had a flow-through system pulling from the reef right onto the beach at our little house there. And we decided we wanted to try to take some fragments of the coral, bring it into the flow-through system so that we can see if that fragmented coral will spawn for us. Well, within 1 day, this was happening. And that was in the same water quality as the reef. It just doesn't like to be handled and fragmented like that. The only palmata in captivity right now was raised from larvae. This is just a little colony grown from one individual larvae. And that's it. That's in captivity right now. So I think if we can get it from the nursery, that they're already established on their own bases, they've been fragmented, will be a much better potential for success moving them into captivity. This is a palmata I grew out from a larvae from Puerto Rico, and that was after a year of growth right there. Also, we know what genotypes of coral there are. He has them all labeled. What parent colony they came from, down to what genotype they are. And then we can go further and identify which clade of zooxanthellae is being hosted. The reason I want the genotype information is when I do my experiments, not only do I want to see which species does better long term, but I want to see if there is variation between the different genetics and if there's a certain genetic strain that does better in the future than the other. And like I said, the fragments are nice and established, so nice bases like that. That's a grow-out facility there at Nova Southeastern. And biosecurity is, I don't have to worry about that. There's a lot of uncertainty on what you would get from other aquariums. This is our, I had to showcase a reef tank from the Pier Aquaria. This is our 450-gallon Indo-Pacific tank. And a lot of the individuals around the country that have palmata in captivity, they've raised them in environments like this. So I wanted to stick strictly to the region specific to where these corals came from. I didn't have to worry about the different clades of zooxanthellae and bacteria and whatever that's associated with Pacific reefs, potentially. You can see, I put this picture up here again, you see there's a nice green hue to this coral. He was raised in a Pacific tank, and these corals are naturally golden brown, but we've noticed when you raise them in a Pacific tank, they get this bright green fluorescent color. And then we've also found after if you give them more time and you introduce them into a tank with Caribbean species, they'll get rid of that Pacific strain and bring in the Caribbean strain. All right, so getting into the climate change fitness comparison study, we'll have to work out how to replicate future environmental conditions. Like I said, I'll use my nice little calcium reactor, CO2 reactor to help me with that. There's going to be a lot of adjustment with that. I'm using an Apex aqua controller. I'm sure some of you are using that. But I'm also complementing it with this nice Ocean Optics Jazz station, which is a spectrophotometric pH monitor and data logger. And it will actually measure pH down to 0.0004. So I can look very specifically at how my pH compares between the two controllers and use this as my fail-safe for making sure that I've got exactly the right pH so that I don't get the scrutiny once I publish whatever I do by the super chemists out there. And this little unit, that was a nice little $10,000 spectrometer. So hopefully one day spectrometers will become more economically feasible because they are the absolute most primo precise method you can use. There's a lot of uncertainty with pH monitoring with your traditional electrode probes. Not only with the probes, but also how the probe is calibrated, what calibration fluid you use. The lab next door to me, the Ocean Chemistry Lab, they just published a paper on the variation in just the manufacturers of calibration solution. But there was a lot of variability between the calibration solution that's provided to you. So they've come up with a way to purify the most pure calibration solution there is and use the spectrometer technology so you've got the ultimate precise reading. So when you're talking about little adjustments on the logarithmic scale of pH, you need to be pretty precise. And then I've got my nice light meter and flow meter there that I can use once I want to look at the couple effects of thermal stress and light stress and ocean acidification. And then I'll look at the hybrid vigor theory to see if that hybrid actually is more fit in the future. And then the way I can allude to if these corals are being affected or not is the buoyancy weighing technique, rather than just measuring them with a ruler or calipers to see how much linear extension they're having, which I will measure that too. But you actually weigh them in a known volume of water so you can see how much mass they're putting on internally or not putting on. And that way you can get to the bottom of how well those corals are doing compared to the ones in the control situation. And then I can take it a step further, and I've learned how to use a scanning electron microscope, and I can put them under the scope and look at real fine detail of their skeletal structure. These are shots that I took earlier this year to kind of practice, and so that I can see if there's any structural skeletal differences between my control specimens and my treatment specimens. And so you can get incredible detail with the scanning electron microscope. Get you a nice little core light there. And then you can hone in on certain little structures, the split screen. And then you can even make a 3D structure out of it. If I had 3D glasses, this would look like a 3D coral to you. And so that's pretty much the gist of what I do and what I plan to do. I'm just getting to the point where I'm starting to play with the ocean chemistry methodology in the tanks, getting my coral stock set up so that I can really get into the thick of this. And I've just got the survey finished, and I'm compiling that so I can get my master's portion out of the way so I can focus on this big portion later on. And with that, thank you guys very much, and specifically the Reef Club and Madison Speaks for helping put Murray and myself up here. And I'd be open to any questions you have, so fire away. Thank you very much. Yes?
You said before that people think they have enough flow and compared to your flow meter they didn't. How do we find a graph to see where we should be? What would we have?
Well, it's not exactly where you should or shouldn't be, because it really comes down to the combination of water flow with light level and water temperature. So there's not really a set water flow pattern or velocity that you should be achieving. But what I'm just showing is what levels, number-wise, have been associated with successful coral aquaculture. And it's just something to keep in mind, that water flow could be pretty underestimated in your systems. And sometimes when you're having a problem with your corals, a lot of people don't think that it might be a water flow issue, because one thing is you measure water flow in the middle of your tank or near your coral, but you get down even lower to your coral, which I can't get that low with that instrument, it's too bulky, but there is a layer above your coral. As the water flow is going over the coral, there's this boundary layer and friction also with all your branches. So you think the outside of your coral is getting this great water flow, but it's doing its job by slowing that water flow down. So the other side of your coral, or even in the middle of the coral, is having a totally different experience than the outside of your coral. And that water flow is crucial with gas exchange and getting rid of waste and helping that coral stay nice and healthy. So it's just kind of a proxy for what you can think about in your future planning. Yes?
What are your ultimate goals? Would it be to reintroduce the Palmata and Silvercorus back into the wild, or to recreate the reefs through the hybrid?
Well, there's a few different things that I want to achieve. One is I'm very particular to the hybrid. I'd like to get a little bit more attention put towards the hybrid research. There's only a couple of us that actually have put energy into studying it, and I think there's a lot of potential in that species. They said, right now it's only an F1 hybrid, and but it can grow asexually by breaking a branch off, growing another coral. But one of my colleagues, Nikki Fogarty from Nova Southeastern, she's found out that they are producing egg and sperm, and they can back cross with their parents. So what's the given amount of time that it's going to take for this coral to speciate, where it can actually start reproducing among its own species, to create its own species? So I just want to bring a little more attention there. The other big picture achievement is providing the information associated with the impacts of climate change on these species, to provide this information to our federal government and constituents associated with management change, so that they know that this is a serious matter and that there needs to be more efforts put in place, and a little bit of a broader spectrum of approach to keeping these corals around longer term. The other is with the survey and starting the species survival plan in aquaria, that's the other goal, is to be able to start this effort in keeping stocks in aquarium zoos, to be able to outplant on the reef. Or even at least just to start backing up the nursery practitioners. Ken Niedermeyer in 2010 lost almost 3 entire genotypes of staghorn coral during that cold snap. If we had that backed up in aquaria, we could have then provided that genotype back to him. He could have learned from that occurrence. And now he needs to put those corals into a different area for grow out, both in his nursery and on his outplanting site. There's a lot to be considered on those methods, because he essentially has them on a level playing field on his line nurseries and on his concrete platform. So there's a lot of variability in the fitness between the different genetic strains. Yes, Murray?
Do we have any rough idea of the geographic distribution and range of the hybrid?
Yeah, it has been found all the way down in Curacao, which I visited Curacao to locate these species that my colleague Nikki Fogarty originally mapped, because up in the Keys and Dry Tortugas, which I'm working on a permit to get this hybrid from the Dry Tortugas, because there's a stock of it there, a nice healthy stock of it. It's very difficult to go through that permitting process. So if I run into some major red tape, I wanted to have a backup plan. In Curacao, they're not that stringent on their permitting. And also, where those corals were in Curacao, the Curacao Seaquarium is right there off of the seawall. And they have tanks set up for aquaculturing elkhorn and staghorn coral, so they could be kind of my little intermediate there for bringing them from the field into their tanks and recovering those colonies and then shipping them off to me. So they're found all the way in the southern Caribbean, all the way up to Belize, and they're found in the Florida Keys. Haven't heard of any up in South Florida yet, but it's all on the matter of who's out there looking and can tell the difference between these corals and report it. So there could be more of a broad range of that hybrid than we think. Anybody else? All right, well, I'll be around, so feel free to come up anytime and be glad to talk with you. Thank you guys so much for your hospitality.
All right, we're gonna take just a quick 5-minute break. Let people go to the bathroom, grab a fresh drink. You wanna,
We got it,
come up here and get a high five.
We got it.
Whatever, we're all about it.